know, you mentioned horizontal gene transfer. Would you be able to talk about what exactly that means?
It would be the transfer of genes from one organism to another organism. And in bacteria, this occurs rather frequently. So we have the phenomenon that perhaps many of you are familiar with of multiple antibiotic-resistant bacteria. And a bacterium can pick up the resistance to several antibiotics in one fell swoop, one transfer of DNA through a unit we call a plasmid from one organism to another. And this, of course, can be very dangerous for human health because all of a sudden an organism that you didn't think was a problem could be if it's pathogenic, and you can't use antibiotics to treat it.
So
I'm not really the specialist on this. Rita's probably better than I am.
But it's interesting to think, because we think about things being transmitted from the parent to the child. I don't know whether you say parent to child about bacteria, but that it would be one generation to another.
Yeah.
But what you're talking about is organisms of the same generation sharing their genetic material and changing in that generation.
That's correct, and it's a pretty surprising thing because it does seem to happen. It's more frequent between organisms that are very closely related to one another than those that are farther apart. But there are examples that people have claimed of inter-kingdom or even inter-domain transfers between the bacteria and the archaea, for example, which seems quite remarkable. And this, I think, is something we do not yet fully understand because of the vast diversity of bacteria. It's sort of ironic, let me mention this one thing, that we know there's like 1 million animal species, and we showed you how diverse all these bacteria and archaea, but you know there's only 5,000 or so named species of bacteria. It's pretty remarkable.
That's the 1%.
pretty sobering.
Yeah. Rita, Jim mentioned antibiotic resistance. What does this mean for public health and for the way that we deal with disease?
This is a very, very serious problem because, well, I think it goes back to the mid-50s when the Surgeon General of the United States announced that we didn't have to worry about infectious disease anymore because we had antibiotics and, you know, we just take care of them and we should move on to other things. Well, what has happened is that we've very, very inappropriately used antibiotics. For example, we use tons of antibiotics to mix with feed for chickens, for cattle. We use antibiotics to treat virus infections, which is absolutely senseless because it doesn't have any effect whatsoever on a virus. So we've ended up essentially strewing the environment with antibiotics, and as a result, the microorganisms have developed resistance. They have modified their genetic makeup. They've either picked up genes that allow them to bypass the antibiotic or to metabolize it and break it down so that they're resistant to it. And now we end up with almost our entire armamentarium of antibiotics being ineffective because the resistance genes to those antibiotics, as Jim pointed out a minute ago, can reside in a single bacterium. So if then you have a multiply drug-resistant TB, tubercle bacillus, this can be a very, very serious public health problem.
Are there things that we can do, and I'm thinking about the non-scientists in our group. I mean, in the stores you can find antibacterial soaps, you can find things that you can use to kill just about anything. Are there things that we can do to make sure there's more of a, I don't know whether it's more of a diversity, but that we can avoid this kind of resistance happening?
I think part of it is just simple hygiene is really very, very important. I think, at least in this country, you do need a prescription. In a lot of countries, in Latin America and some countries of Europe, you can walk into a drugstore and you can buy any antibiotic, even those that are dangerous. Some that, for example, there is an antibiotic that causes aplastic anemia. You don't want to take it unless every other antibiotic hasn't worked and that is effective. You don't want to be taking it. So I think in the 21st century, with the ability to Google on the internet and find whatever it is you need to know. I think it is important for the individual to be informed. The best defense is to be informed.
You know, you mentioned hygiene, and I think most of us, our first experience of bacterium, of microbes, is the ones that make us sick. But are there ones that we need to stay healthy, too?
Actually, the number that make us sick are really minuscule in view of the entire microbial underpinning of what we call planet Earth. We really are sort of glued together with microorganisms. And if we didn't have them, I think we'd essentially sort of fall apart. I mean, that's a little dramatic, but nevertheless, there are many, many more microorganisms that provide fertility in soil, that provide, I guess as it was pointed out today, we are 1% human cells. Was that the rest is microbial cells?
In our own bodies.
In our bodies. So we're walking microbial populations. So there are many, many beneficial microorganisms, and I think...
Yeah, maybe I can just add a little to that. Think of it, the tree of life also shows that plants and animals evolved more recently, and that's consistent with the fossil record as well. That means that the plants and animals evolved in a world that had been occupied by microorganisms for 3 billion years.
Before the plants and animals
Before the
showed
plants
up.
and animals came about. And what the plants and animals did is they made friendly associations with the bacteria and archaea and so forth, and in particular the bacteria. But we have in our intestinal tract, because of that, the bacteria growing anoxically, anaerobically, without oxygen, which we can't, our own cells cannot do that, but these bacteria do that, and they're essential to us for the breakdown of our foods to provide us with amino acids and vitamins and so forth. So.
I know, Susan, that Dr. Novacek mentioned the idea of obesity and gut microbes. What's been happening there that we now understand about these colonies of microbes that are inside us?
Everyone's getting yeah,
And
so this just happened fairly recently. So some work done by a postdoc named Ruth Lay and some others. They started to look at people who are obese and people who are lean and just look at the species of microbes that they had in their guts. And immediately you saw two very different profiles. And then what was really interesting was they took people who were overweight and they put them on carbohydrate-reduced and fat-reduced diets for over the course of a year. And they watched as the microbial population shifted over that course of a year. And so it's just a fascinating thing to me that a person's weight, a person's appearance could be tied to the microbes in their gut. And I'm not sure that it's known right now whether it's cause or effect. So if you're a lean person and you're exercising and you're burning a certain number of calories and metabolizing, you're selecting for this group of microbes. And if you're sitting on the couch and using the remote control all day, you're selecting for a different kind of microbes. Or if the microbes are saying, sit on the couch and flick the channels. So I think this is really exciting research to see who's controlling whom. And it could open up some really interesting doors for diets, as Dr. Novacek was saying in the beginning.
You know, I was just remembering the cover of Discover magazine a couple of weeks ago that they now think that autism may be connected to our gut microbes as well. Rita, do you know anything about that research?
Well, it's tenuous, but it's fascinating. I mean, I've learned over my career to be very, very tolerant and open-minded to new theories, because some of them turn out to be quite true and very astounding. But the notion is that we are susceptible to the kinds of microorganisms that produce compounds that may influence behavior. And I think it was fascinating to learn the other day that it is our small intestine that has a very, very large amount of neural connections and ends up controlling or at least influencing behavior because of the compounds that make their way from the gut and the signaling that makes its way from the gut to the brain. So it's a fascinating kind of area, I think, of research. I think the next decade is going to be really very dramatic. Do you have any comment on that, Susan?
No, I think that's true. David Relman this morning was talking about one of the roles of microbes, which only recently has been discovered, is helping educate our immune systems. So you hear a lot about if you raise a child in a very sterile environment, it opens them up to disease, and we think, oh, it's just a matter that they're not exposed. But then it's even more complicated because they're not exposed to the good things that start training our immune system to function correctly, so you don't come down with allergies and other autoimmune disorders. So yeah, it's fascinating.
It is fascinating.
But the other thing I would mention is that it was emphasized by a person, not at this meeting, but at another meeting earlier this week, that the assumption is that what we ingest, we calculate the calories, we calculate the nutrients, and the assumption is that that's incorporated. Well, maybe not. Depends on the microorganisms. Did they actually break it down so it could be incorporated or not? So it's a very interesting area of research.
You know, we've been talking a lot about how we depend on microorganisms. Jim, you had mentioned briefly how plants and other parts of the earth depend on microorganisms. I know many of us who have gardens or window boxes are about to plant things for the spring. How do plants require microorganisms in the way that we do?
Yeah, the plants usually have microorganisms associated with their root systems. Some of them will even have microorganisms in the plant tissue as well. The other thing are these great cycles of nature that the bacteria and archaea carry out, and it's probably worth mentioning them at this point. The microorganisms are the greatest recyclers on Earth. They take your garbage, they break it down. The sewage waste, they break it down. So these carbonations, the wood in the forest, the leaves, all of this is broken down by microorganisms and so forth. Animals play small roles in some of these things, but by and large it's a microbial thing. And we think of these cycles occurring, and I don't know what they have. Yeah, we have the nitrogen cycle here, and let me just talk about that a little bit. So plants require nitrogen in the form of either nitrate, which they can use and produce amino acids from that, or they can take nitrogen in the form of ammonia. So those two arrows are what plants can do. Animals, mammals in particular, require amino acids. So this amino group here is from an amino acid. And they can get that amino acid from eating plant material or other animal material if they're a carnivore eating another animal. But all these other arrows here are carried out by bacteria and archaea. And these cycles have been going on Earth for literally billions of years. So you have special bacteria that oxidize ammonia, and just recently, just a year or so ago, the first archaean that carries out this what we call nitrification process was actually isolated. So they're both bacteria now and archaea that can do this. And I think Jim Teege alluded to this AMNH reaction. It's carried out by a special group of bacteria called the Planktomycetes. This wasn't even known about 10 years ago. And you can see it's changed the nitrogen cycle completely. It uses ammonia and, as an energy source, and this actually, there's an error on this. It should be nitrite here that it uses also. These two nitrogen sources, one of the energy source, this is oxidized, the electrons go to the nitrite, and out of that you get one molecule of nitrogen gas, one atom from ammonia, one atom from nitrite.
And is this something we can, once we learned that plants needed nitrogen, could we then manufacture this and not depend on the bacteria?
To some extent, but it's very, very, very short term. I might say nitrogen fixation, where you take nitrogen from the atmosphere and fix that into amino groups, is only carried out by bacteria and archaea. So it's another, and that's an essential process. We need to have a balance here between the nitrogen that's that's fixed in this manner, and then the nitrogen that's lost through this Animox reaction and the denitrification. So this is all balanced, and this stuff has been going on billions of years, and it's something that we can't really change. It's like the tides. They are occurring. We can't stop them, and thank goodness maybe we can't, but we can influence them. So as global
Both for
temperature,
good and for bad.
for good or perhaps for bad. For example, as global warming increases, the rates of these reactions go up. For roughly every 2 degrees in Fahrenheit, there's a 10% increase in rates of microbial processes. So there are concerns about global warming effects on microorganisms.
Rita, you gave a presentation this afternoon where you talked about cholera and its interaction with a very tiny creature called a copepod. And I found it fascinating because you were not just looking at these two creatures and how they interact with each other, but looking at water temperature and ocean height. How did you come to think that those things might affect the organism you study?
I think it's necessary to take a holistic view. I like the words of Muir, John Muir, who was the founder of the Sierra Club. In 1848, he said that when you tug on any one thing in the universe, you find it connected to everything else. So we've been in a very productive reductionist period of research in science for the last 75 years.
And by reductionist, you mean going
Smaller
and smaller.
going down to the molecule, to the electron, to the forces that make things happen, when ultimately we have to put this together to understand how it works. I mean, you can take the clock apart, but you need to put it back together if you're going to tell time. So I think that understanding an infectious disease, for example, means that you don't just discuss and work with the patient and the physician. That's just one small part of the whole complex of the environment, the nutrition, the season, the climate, the infectious agent, its state of infectivity, your, as a host, your state of receptivity to the infection. It's a very complicated process, and I think now with the capability of the high-speed computers, information technology, the kind of modeling that's done to understand the cosmos through astronomy, we can take those kinds of principles and we can begin to understand how things function here on Earth. And so that's what led me to this more complicated, integrated view of a very important disease, cholera, which is an organism that really is a naturally occurring bacterium in the environment. We can't eradicate it because it plays a very significant role in recycling nutrients in estuaries and rivers and coastal waters. But it also has the capacity to infect us if we ingest it. And it's understanding that process. We had a very nice discussion today from Dr. Dobson about parasites and how we are an inappropriate host for one of the larval stages that makes us itch, doesn't make us infected. Well, the cholera bacterium, in my view, after 30 years of studying it, I think the bacterium, when it attaches to our gut, in my view, it reacts as if we are a large copepod, and it's trying to help us osmoregulate. It's trying to help us tolerate the changes in salinity, and it just interferes with the metabolism for sodium-potassium transport, and it makes us really sick. We lose a whole lot of fluid, up to 18 liters in severe cases of fluid, through diarrhea and vomiting, and through the systemic shock, death ensues. I don't think it's primarily a pathogen. I think it's one of those situations where it's carrying out its natural function.
That if we were copepods, it would help us,
It
but
would
we're
help
not
us.
copepods.
That's right.
One of the things I found inspiring, too, is that through your research and your understanding of this interrelationship between the cholera and the copepods, you came upon a very simple way for humans to try to keep the cholera where it can help the copepods and not in our own systems.
Well, that was because I've worked in Bangladesh for almost 30 years, not full-time, but doing research with my colleagues there. And when you are in a cholera hospital with, I remember one very bad cholera epidemic where there were 1,000 patients per day for a month coming into the hospital, you really want to help people. And I think that's really fundamentally what drives scientists, engineers, technologists. It is really to improve the lot of life on this planet. And so with this simple hypothesis, namely having done all this basic research with elegant tools, being able to understand this interconnectivity and to put a barrier between the pathogen and the host, namely by filtering it out, it's very rewarding to be able to have been able to help people.
Can you describe the simple approach that you took?
Yeah, yes. The bacterium is attached to the copepod, which is, by microscopic standards, is really an elephant. It's about 280 or 300 or even larger microns, micrometers. The bacterium is only about 1 or 2 or 5 micrometers. So sari cloth, we tested a lot of different cloth because we wanted to get a really inexpensive filter to filter out the plankton. And we tried men's t-shirt material. It wasn't any good. We tried Chinese poplin. It was expensive and it was no better. But we found that if we folded sari cloth, old sari cloth, because it was frayed and it really gave very small pore filter, it was like 20 microns, 18 to 20 microns.
And this is the clothing they were wearing in
It's
Bangladesh.
the clothing they were wearing. And then there's a funny story to tell. When we proposed to the NIH that we use this sari cloth where you could fold it 3 or 4 times, filter the water, then you'd unfold it, rinse it, dry it in the sun, and the sunlight would act as sort of a disinfectant. One of the reviewers said, men will never drink any water that's been filtered through a woman's used sari cloth, unclean. Oh. So we did a pilot study for 3 months, and during the study, we found out the men had been using sari cloth to filter the flies from their beer.
But through this very simple approach, you were able to reduce cholera transmission.
Indeed. Yeah, we were able to reduce it about 50%. And we've been very heartened because in a study we're carrying out right now, we find 5 years later that 70% of the villagers are continuing to filter their water through sari cloth, and we are finding that the cholera rate is the lowest it's been in many years.
Now, we've been talking about bacteria that we interact with. Jim, one of the things that you look at is bacteria that live in these extreme environments in polar ice or in thermal vents, places where other creatures wouldn't be able to survive.
Right. Yeah, our own work is more on the polar ice, but many people are also working on these hydrothermal vents, which are very hot environments. I guess the major point to make here is that the bacteria live in environments with higher temperatures than any plant or animal could live in, and also at lower temperatures than any plant or animal can live. And they also live at low pHs, very low pHs, and high pHs, and other very low water activity or humidity environments as well, and on and on. So the upshot of all of this is the bacteria, you can think of, they've evolved for these billions of years, and they're in environments that no other life can be in on this earth. And so they're in all ecosystems, whereas plants and animals are not. Now this is important for astrobiology, because we know, even in our own solar system, that there are extreme environments that may contain life.
And that's what astrobiology means, is life beyond what we know of as this Earth.
Well, that's what exobiology is.
Okay.
Exobiology, the term was replaced by astrobiology because exobiology talked about life outside of Earth, but we don't know of any life outside of Earth. Astrobiology looks at all life in the universe and asks the big questions: are we alone, or is there life elsewhere also, this sort of thing.
There was a lot of excitement about that this week with the notification that they found a planet that they think life might be able to live on.
Right, yeah, this is a recent finding. It's been a major focus of astronomers for the last, about 5 years. What they're trying to do is identify planets that are fairly close that could be in what we call the habitable zone. The habitable zone is a zone near enough to the sun to get energy and warmth, but not so far away that there isn't water, for example, or liquid water and that type of thing. So in this particular report, they have found the closest example of a planet that's like the Earth. It's not exactly like the Earth. It's about 5 times the mass, and I think 1.5 times or so they figure of the diameter and that type of thing. So it is a possibility that there might be life on planets like this. And it's 20 light years away, but it's not.
It takes a long time to get there.
You're not going there tomorrow.
I'm not going there tomorrow, but maybe someone else. Actually, it's not too far away from an astronomer's point of view. So.
Actually, Jim, I like the literal translation of astrobiology. It's life amongst the stars.
Oh, that's
that's good too. Carl Sagan said there's billions and billions of stars. Well, we've got more microorganisms than there are stars, so we've got a lot in common here.
We're going to be taking your questions in just a minute, so if you have any questions and you haven't already passed them to the side, please do, and members of the staff will collect those and we can take a look at them. One thing I wanted to ask you, Susan, about is there was this recent article in National Geographic, which I found fascinating, about the Jamestown colony. And it began with a fact I had never known and found rather startling, which is that there were no earthworms in this part of the country before the settlers came, and that they brought over in the soil that they used for their ballast earthworms that then colonized the States. Now, these are creatures we can actually see. Do similar things happen on a microorganism level, too?
Yeah, so on the same ship, the Europeans coming over to colonize Jamestown, and a lot of the other European colonists were bringing microorganisms of their own. So one of the most important things they brought, and one of the things dear to my heart, was malarial parasites. So until Europeans came along, there wasn't malaria in the New World, and it's actually suspected that bringing some of these diseases, not just malaria, but things like smallpox and other diseases, might have given the Europeans a bit of an advantage and allowed them to take over. So we certainly know about invasive disease organisms. We don't have a clue about some of the other organisms. So you mentioned the soil coming over from ballast ships and introducing earthworms. What were the other microorganisms that were living in that soil and how they changed? We have no idea, and I'm sure we never will. One of the things we've definitely learned today is that microbes are not ubiquitous. It's not the everything is everywhere, but there is a lot of biogeography. So we know that the Columbian expansion in the New World changed biogeography substantially, both old world to New World and vice versa. I don't think we are even at a tiny fraction of the point of understanding how microorganisms also changed in that period of time.
I know, Rita, in the slides you showed today, too, you talked about dust plumes from one part of Africa going to another and containing microorganisms.
Well, it's suggested that the incidence of asthma may in fact be related to the dust plumes that come in to Europe from the Saharan desert and from the western part of the United States from the Gobi Desert and the storms from the desert of China. So the evidence is pretty clear that there are bacteria, fungi, and quite likely viruses carried in those plumes. And Most of them are probably harmless, but some may not be.
Before we go to the questions from the audience, one of the things that I found fascinating is that we're actually harnessing the power of microbes now. That there are people talking about microbes that can turn garbage into energy or other things that can clean up oil spills. In your explorations, what have you heard about or touched upon that you find really promising going forward?
Well, there's a lot of interest now in ethanol as a fuel source, and the production of ethanol from corn is ongoing right now, and you probably know that Brazil is no longer reliant on fossil fuel for its oil, uses the ethanol from in this case from sugarcane from Brazil as their source of fuel.
And is ethanol a bacterial byproduct?
Yeah, it's a product of the yeast that was mentioned earlier.
So beer, pizza, and ethanol are all things.
Yeah, and the future is to look at cheaper sources because corn is rather expensive. That's what's used in our country, which is corn starch. But the use of cellulose, wood materials, and bacteria break down cellulose. They have cellulase enzymes that will do that. And so there's a lot of research going into that area right now.
Well, we have a Prius. My husband's very proud of it. He drives past the gas station and thumbs his nose. But I would like to see the power of biotechnology employed. I really don't think ultimately converting farmland for food production, from food production to production of fuel is really the ultimate answer. I think it's learning how to take the kinds of organisms you've been working with and some of the discussions we've had today to splice genes and produce an organism that can provide the kind of energy. For example, if we simply harnessed the sewage treatment plants, we're discharging sewage, but that's an enormous source of energy. The methane that's produced is burned off, and if that could be cycled, we could generate electricity.
We've denigrated and condemned you, not knowing we're in the same stew. This cosmic kettle of life on Earth is filled to the brim with you. Bacteria, chugga chugga. Bacteria, bacteria, chugga chugga. Bacteria. It's time to appreciate the microchemistry that is the key to our sustainability. Billions of years before we were born, microorganisms sang the song. Bacteria, chugga chugga. Bacteria, bacteria, chugga chugga. Bacteria. Humans think we're at the top of the heap, yet we never ever could be complete without these tiny creatures that make up our form. We pay them tribute as we sing this song. Bacteria, chugga chugga. Bacteria, bacteria, chugga chugga. Bacteria. 3.5 billion years ago, bacteria came forth to join the show. 8 million years later, crisis arose. Imagine, oxygen was the flow. Here's how the story goes. Oxygen was a poison long before we had a nose. Bacteria adapted, learned how to breathe it. Because of them, we are here to sneeze it. Bacteria.